Greetings. My name is Anna Zimmerman. I teach immunology here at the College of Charleston. And today, my good friend, Dr. Phil Dustin, has asked me to do a guest lecture for his ecology class concerning COVID-19. So, scientists are always learning new things. Here's a picture of me when I went halfway across the world with Phil to go scuba diving in his ecological world in Indonesia in Raja Ampat. And fair is fair. So here's a picture of Phil when I took him down the hall at the college to show him how to isolate DNA. So today, none of us are getting to travel anywhere, and we are under quarantine. Quarantine has an interesting history over time. We certainly have not been the first people to be under quarantine, and we will not be the last people to be under quarantine. And if we're finding a couple weeks being under quarantine difficult, go ahead and Google Mary Malone, AKA also known as Typhoid Mary. What you'll find is she has one of the most fascinating cases of public health and quarantine, in that she alone was quarantined on an island due to her spreading typhoid fever. And this quarantine for her lasted over 20 years. So while it may seem that what we're dealing with today, this virus came out of nowhere, viruses really don't come out of nowhere. And scientists have been studying viruses for decades. And what we have seen is that we have had some epidemics or outbreaks on a localized level of viruses that are other coronaviruses. One being the SARS outbreak that we saw in 2002. And that was an epidemic limited to geographical places in the world. In 2019, we saw H1N1 influenza. And then in 2012, we saw another outbreak of a coronavirus. And this outbreak we called the Middle Eastern Respiratory Syndrome coronavirus. And that was also an epidemic. What's different in what we're seeing today with our novel coronavirus is that this has gone from the state of being an epidemic, which is a localized area of infection, to a pandemic, which means it's a global distribution over many different geographical areas. And pandemics are going to have a large ecological component. Ecology is going to be intricately mixed with pandemics. And what we're seeing is that human health, animal health, and the state of ecosystems, they really are linked together. 70 to 80% of the emerging infectious diseases are known to be of zoonotic origin, meaning that they can be transmitted between animals and humans. There are a lot of viruses that are circulating in animals that have yet been found to be infective to humans and vice versa. And we know that with the changing world, with population growth, climate change, increased urbanization, and international travel and migration, what we can see is that we can have a risk for the emergence. And what we're seeing on a global level is a spread of respiratory pathogens. So we can all probably remember when we've heard about these avian influenzas or bird flus that have gone around. Maybe not so much have we heard about the most recent coronavirus outbreak that happened before what's going on today. And we call this the MERS coronavirus. And this was very interesting that this happened in the Middle East. That's why it's given that name. And we had about 2,500 cases total globally that happened, and about 850 people died. So the death rate of this was 34%. And what was interesting about this one is it was thought to be transmitted originally from camels, and camels were able to fight this off pretty well, much better than the humans that were infected with this virus. So MERS and SARS, these are types of coronaviruses, as is the virus that is causing COVID-19. Why we call them coronaviruses is due to their physical appearance. And if we look on the surface of a coronavirus, what we see is we see a lot of these proteins that are extending out on the membrane. And we call this a crown because it kind of looks like a crown. And so, crown, corona, that's where it gets that name. So, coronaviruses have been found to cause disease in a wide variety of animal species. And what we have in the situation of a infection that can go from animal to humans is we call this a spillover event. And a spillover is basically what happens when we have a situation like maybe our coronavirus in a camel and something makes it possible that now this can infect another species. So we call that a spillover. So viruses and the diseases that they cause are often named different things. So much like we know that HIV is a virus, HIV causes the disease of AIDS. So people don't die of HIV per se. We would say they died from complications arising from AIDS. And in the case of our novel coronavirus, that has been named SARS CoV 2, and that stands for Severe Acute Respiratory Syndrome Coronavirus. And dash 2 makes it different than what we would call what was originally corresponding to SARS. The disease that this virus is causing is has been named COVID-19. And what I would recommend is when we're looking through our information, really our best statistics are going to come from the World Health Organization, the Center for Disease Control and Prevention, and the NIH. And looking at some of the data that's coming out of these organizations and the estimates that are coming out of these organizations, they're estimating that maybe this will be asymptomatic in 80% of the population. And it does appear that for some people, symptoms are developing. And some of the symptoms that have been attributed to this virus causing this disease have been fever, dry cough, shortness of breath, headache, sore throat, and muscle aches, and possibly some other non life threatening symptoms as well. What's been a little bit confusing about the diagnosis of this is it's been difficult to diagnose something just based on these symptoms. And even more confusing in some cases is that some people might exhibit one or two of these symptoms, somebody else might exhibit all of them, and the range in between. So, where this COVID-19 gets real is going to be in the lungs of the human body. So, what we've learned from SARS is that these coronaviruses can cause very detrimental effects to the human lung. And the most serious is going to be when we have a collapse of basically these regions in the lung that we basically can call the air sacs of the lung. When we have a situation where these air sacs become compromised, people are in a world of hurt. And there's a lot of evidence emerging that we have a pretty robust immune response that's going on, but sometimes the immune response is not going to be strong enough, or it's going to have side effects that can be somewhat detrimental as well. We've seen evidence now with the virus that causes COVID-19 that we have a massive infiltration of B cells, macrophages, T cells, and also some really cool cells that are called neutrophils. So much like chemists have the periodic table that kind of guides their world, immunologists have lineages of hematopoietic stem cells. So basically, stem cells and the differentiation of these cells down different pathways is going to be like the periodic table for the immunologist. And so here we have a beautiful depiction of all kinds of cells. I could talk for days on what each one of these cells does and very important roles that they play. But since we're talking about the coronavirus in this situation, let's just get to the point of those cells. So I said that neutrophils can play a role. Well, neutrophils are going to be cells that basically can release granules, and neutrophils are going to be really important in the formation of pus. Also, B cells are going to be the cells that go ahead and differentiate into something called plasma cells. Plasma cells are awesome in that they secrete antibodies, and our T cells are going to be very important role in helping out B cells to make those antibodies. And T cells also are going to have an important role in killing virally infected cells. Straight up, a CD8 positive T cell can kill a virally infected cell. And then macrophages are going to be really important because a macrophage, we can think of a macrophage like Pac Man. And if a virus gets coded by antibodies that are specific to that virus, a macrophage is basically going to be able to eat it up. So many ways we can think about a macrophage is sort of like Pac-Man, and a virus is sort of like a French fry. If the virus gets coated with antibodies, and we can say the French fry is coated with ketchup, that gets it really tasty for a macrophage to come in and go ahead and gobble up that virus and destroy that virus before it has a chance to go and infect other cells. So, immunologists all over the country are making these really cool diagrams to show all of these different cells in action, how we can have our coronavirus, we can go ahead and make antibodies against that. And we could spend the rest of the lecture talking about what's going on in this picture, but that will be left for when you take immunology, hopefully with me next year. So, the respiratory epithelium, if we wanted to look at a little bit of anatomy and physiology, here we know that we all breathe in air when we inhale, and that air is going to go into our lungs. Well, what's pretty cool about our lungs is our lungs are made with a really cool lining of cells that we would call the epithelial layer. And those are the cells that are lining. We have cells like depicted in this little um, square here, and we also have epithelial cells in the lungs itself. So, what we have been learning is that the SARS-CoV-2 virus is going to be infecting these epithelial cells. Now, once infected, the virus can hijack the machinery of the epithelial cell and make copies of itself. And then those copies can be released. The cell could burst, and we could have a situation where the virus then is going to infect all of these adjacent cells. Well, our immune system, typically, when a cell is virally infected, we actually are going to have T cells that can come in and go ahead and kill a virally infected cell. So maybe the cytotoxic T cell, which is called a CD8 cell, comes in and kills these cells. Well, that's great if we want to get rid of basically little factories that are now making viruses, but it's not so great if our body cannot replace these cells as quickly as they're being destroyed or shortly thereafter. And in the lungs, this becomes increasingly problematic because we know that when we inhale into the lungs, we know that that oxygen is going to be transported into the body. So, if we're getting rid of cells that basically are transporting oxygen to the body and we're getting rid of them at a rate that is too quick in order to get them replaced, that can be problematic. What can also be problematic in the lungs is we are bringing in all of these immune cells, and the immune cells themselves, although they're fantastic, they can be causing some inflammation. Tie that in with dead, dying cells that are going on due to the virus either lysing those cells or the virus infected cell being killed by a CD8 cell, and we can end up with a lot of problems with inflammation. And when we start losing these cells in the lung, these epithelial cells, well, these cells also have these wonderful projections that are called cilia. And cilia is going to be great in capturing any type of inhaled particle and basically moving those particles through mucus. Imagine if we get into a situation where we start losing cilia, then we start having mucus accumulation in the lungs. We can't cough up particular material. And this deposition of mucus also has some problems when this starts going on. So, in order to kill the factory, which is making additional virus inside the lungs, the CD8 cells have a job to do at killing those lung epithelial cells. Well, killing those lung epithelial cells, although it shuts down the factories for making more virus, which causes more problems with cells, it also has the effect of decreasing cilia in the lungs. And that's certainly, as we just talked about, is going to impede mucus transport. If we get to a point where we have all these cells dying, we're going to get inflammation. And the end result is that people end up getting fluid buildup in their lungs. And the end result, what we would consider that is pneumonia. So, pneumonia is the big problem here, that people get into a situation where there's fluid buildup in the lungs. And what that means is that their air sacs in the lungs become inflamed, filled with fluid, and it really makes it hard to breathe. So, pneumonia is not exclusive to COVID 19. Pneumonia can be caused by a lot of things. And pneumonia is a condition, it's not pathogen specific. So, we can get pneumonia from bacteria, we can get it from fungi, and we can certainly get this as a problematic effect of viruses, such as the virus that is the causative agent of COVID 19. So tie in together fever, chills, cough, shortness of breath, and pneumonia, and this can get really serious really fast. So let's have a little chat about pneumonia. So we know with pneumonia that the lungs can become inflamed and filled with pus. The pus is going to be brought about primarily by those neutrophils that we talked about, and it's going to make it really difficult for people to breathe. So, needless to say, pneumonia is not fun. I've only had pneumonia once in my life, and it was one of the worst weeks. I would never want to have to relive that. So, certainly, pneumonia, it is no fun. When you have a difficult time breathing, that has a cascade of different factors that it gets really difficult to get oxygen to the blood. And when we have low oxygen levels, we can lead to a situation where we can't get oxygen out into the body, and we can start to see organs developing lots of problems. And the longer this goes on, we see people that start to have organ failure. So these coronaviruses, they can infect the epithelial cells, and then we can get a situation where the epithelial cells work as little factories while we're trying to kill off the actual factory. We try to get rid of the cells that are infected. That has problems because we're removing those cells that are integral in getting oxygen into the blood. And if we are lacking oxygen in the blood, we're not able to deliver oxygen to the tissues, and this can get into a situation where people have serious pneumonia and organs can start to fail. So in the most severe COVID-19 cases, what we're really seeing it's this lack of oxygen that's driving multiple organ failure, respiratory failure, septic shock, and patients need supplemental oxygen in order to survive. And in the most extreme cases, what we're seeing is that patients require mechanical ventilation. So enter the respirator. Respirators add oxygen to the blood and they remove carbon dioxide. So we can see why this discussion of lack of respirators is going to be extremely problematic as we move forward with more people becoming infected. So, what we're seeing is a tragic case of viral victory on the world stage. This is a fantastic site. I got this graphic from the Johns Hopkins University coronavirus website, and I put the address here. This is a great site, and that you can look up some of these statistics that are emerging worldwide. And this is really, really serious. And we're living in historical times. The first person to ever track down a disease was a medical doctor in London by the name of John Snow. And John Snow, oh, where will this go? That's what we're all waiting now. We're all waiting to see where this is going to go. If you get a chance to go to London, and hopefully we'll all be getting chances to travel in the somewhat not too distant future, you can actually go visit the site at which the first epidemiological study in the world was done. And that was done by John Snow. He was able to track cholera and the distribution and sickness that was coming about from this bacterium to this water pump. You can even go on a little bit of scientific tourism. You can visit his grave. They have a nice plaque on this reproduction of the water pump, as well as a plaque on the wall next to where he was working to track this down. So we're living in very historical times right now. And being an optimistic person, I really think that we are going to have some heroes that emerge in this process. And we're going to have people like the Jon Snows of today, which are going to be looked at from the history of immunology. John Snow is considered the father of epidemiology. Maybe we're going to get some brothers and sisters and mothers and fathers that are going to emerge from the current pandemic with some awesome new science to help us get through this. And there are a lot of smart, creative, determined people that are working all over the globe as we're listening to this lecture now. And they're working on a variety of different possibilities, repurposing other drugs, convalescent plasma therapy, where we take antibodies from people who have recovered and we use those as a protective therapeutic to people that are currently infected or maybe people that are immunocompromised. There are folks that are working on trying to make antibodies outside of the human body in order to be able to use those as therapeutics. Antibody assays are coming to market now almost. They have been approved by the FDA at rapid speed so that we'll be able to test pretty soon who has been exposed to this virus and who has recovered. Those are going to be great things. And then the gold standard, which a lot of immunologists are really hoping will come about, but this one takes maybe a little bit longer, often a lot longer, is a vaccine. And much like the world was able to eradicate smallpox, I think that we can look at this as one day we will hopefully have eradicated this and we'll be better protected in the future for these types of epidemics. So, in times of crisis, I tend to be of the mindset that the children are always watching. And with any crisis, we need to be the people that they need us to be. And so, here I'll end with this photo. These are children on Arborek Island. Arborek Island was the island that Phil, Dustin, and I were able to stay on when we went on our scuba diving adventure in Raja Ampat. And if we think about this on a global scale, although this seems so far away from Charleston, we really are at a global situation right now, and we do need to be the people that the children of the world need us to be. And so that can be staying home, that can be not spreading this can be taking this quarantine in stride. Again, there have historically been quarantines that have been much longer in the case of Typhoid Mary that we talked about, and just really hopefully we emerge with the silver lining as being a more compassionate society, a society maybe where science is held in a little bit of a higher regard. And we're going to need people like you. We're going to need scientists in the world. We're going to need creative, informed, capable, passionate people that can go ahead and solve these diseases in the current time and diseases that we're going to encounter in the future. So be the people that the kids of the world need us to be. So I hope you enjoyed our discussion through COVID 19. I appreciate Dr. Dustin inviting me to do this guest lecture. And I'm going to leave you with something that one of our College of Charleston students put together. And so you can share this on her YouTube channel. And I asked Kate if I could go ahead and include this in a voice thread. And she told me to go ahead and share away. I think this is a really beautiful rendition of a lot of what we're all feeling today.